This episode is brought to you by The Wellness Summit, 2 days that would change your life forever. For tickets, go to thewellnesssummit.com. TheWellnessCouch.com, streaming wellness into your lives. This is Up for a Chat with Cyndi O'Meara, Carren Smith, and Kim Morrison.
Here we are, up for a chat about the hottest topics that are important to you, inspiring you to awaken the change within. I'm Carren Smith.
I'm Kim Morrison.
And I'm Cyndi O'Meara.
And welcome. Because we have a special guest, Steve Myers. Welcome, welcome.
Yay.
Well, thank you. Thank you for having me again.
Actually, have we ever had a male?
Oh,
Julius.
we
did have Julius, but he came with his wife.
Yeah, true.
And children.
True.
That,
oh my
true.
gosh, this is our first male by himself.
Are you
Really?
privileged?
I'm a little bit privileged and a little bit scared.
You should be very afraid. Yeah. So Steve has been, or he started with Cyndi, and I'll let Cyndi tell you the whole story about this, but one of the reasons why we decided to get Steve on the show today is because we were doing some recording and Cyndi was saying, what are we going to talk about on our next show? And I said bioidentical hormones, and Kim said diabetes, and we were also throwing all these things out to Cyndi, and Cindy's like, eyes started to roll, and she's like, okay, time to get Steve in. And I think that the story of how Steve has come into all of our lives, because yes, he is in all of our lives now, as of this moment, I think is really quite fascinating and really amazing, because it's a journey of enormous growth for you, Cyndi. So I'm going to shut up and over to you.
You want me to tell the story?
Totes,
Okay.
go, because it's a beautiful story. But then we're just going to smash you, Steve, with our questions that we just have to have answered by a PhD.
Sure, I'm ready.
That's all I know. I didn't even know his name was Steve.
I know, you came in and called him
Mr. PhD.
Mr. PhD.
That's all right, I like that.
That's all I've called you.
Yeah, that's fine.
It has been an incredible journey, and I believe Steve is the perfect person for what I wanted. So I conceptualised you quite a while ago. So probably a couple of years ago, I started to think I wanted to do an education course, but I had no idea how to do it. And I knew I needed somebody that was a lot smarter than me, that had a real thing about intricacy, so they could get into, like, I skim the surface. I don't want to know the detail, just I'd skim the surface.
Really?
Yeah, I just skim the surface.
Wow.
Well, I didn't think I used to until I met Steve.
Because I'm thinking to myself, I've never would have.
Yeah.
No, I speechless. Speechless.
See.
Can't get words out.
But I actually thought I didn't skim the surface, but then when I speak to Steve, I go, oh my gosh, he just goes so much deeper into the whole thing. So anyway, we'll get to that. But so I had this idea, and I didn't know how to do it, and I would talk to people about, who do I get? How do I find them? Do I need to headhunt them? I used to say scalp.
Oh, you did.
Scalp.
I used to say scalp.
You used to say scalp?
Because I had no idea about recruitment. And so
Oh, bless your cotton socks. I'm going to go and scalp somebody. Heavens alive. That's hysterical.
it was. And then somebody would say, it's headhunt, Cyndi. I went,
It's headhunting.
okay.
Oh, you're so funny.
And so they'd say, you need to go to universities, or you need to find someone from the pharmaceutical company. And I'd go, I don't want anyone from a pharmaceutical company. And anyway, what ended up happening is that we did find somebody, but she didn't have a biomedical degree, and I just felt that she was going to have to spend a lot of time getting information that would take years and years and years. Would take years and years and years. I'm showing Steve how they don't even listen to me. They're there on their phones. I know you are. I know you are.
Facebook page.
I know. I know you are, but it's just so funny the way I'm chatting away and Steve's looking at you too.
Sorry.
This is what our Up for a Chat's
I'm
about.
just seeing if there is a Facebook page. Carry on.
Carry on.
There is, but I don't use it very often. LinkedIn is probably
LinkedIn.
LinkedIn's
that's what
probably
I
the
was
better
looking
one.
for.
Hello.
LinkedIn.
Yeah.
I would
No.
have and I would have expected LinkedIn for a professional.
That's true.
LinkedIn and ResearchGate is another one.
ResearchGate.
Yeah, it's very big at the moment. ResearchGate
I'm onto
is...
that. Look at
these girls.
Look at that. Research
Is that where
gate,
you
that
find
sounds like
people,
my
or is
kind of-
that...
Yeah, that's where you can find people, and you can find out, how many times they've published, what their work is about, and things like that. So
And it's a network.
research, it's a network as well. Research, LinkedIn is kind of very broad, whereas ResearchGate is more for researchers.
ResearchGate is a
intelligent
network dedicated to
The intelligent types, yeah.
I probably won't fit in.
I'm sure you will, Kim.
You're so sweet. So carry on.
Oh, so carry on.
So yeah, stop knocking us, telling you that we're not listening. We are listening and researching, actually.
Yeah,
I think it was just so funny the way I feel like I'm talking just to Steve and I'm talking about him. But anyway, so I rang my recruitment girl and I said to her, it's not good enough. She's not the right person. She is if I put somebody who's got a degree, and that's what I was doing. I was trying to combine two people that had all the information that I needed. So Sally came to me and we
Have
sat
you spoken
down.
about why you want this person?
No, probably not, but we'll talk about that. Yeah.
okay.
So Sally came to me and she said, all right, so let's write down all the characteristics if you want from this person. So we wrote down this huge list and she looks at the end of it, she goes, well, you don't want much.
See, aim high.
Yeah, I did. I aimed high. I said, these are the characteristics I want. This is what I want them to be able to do. And she just went, I don't know if we're going to get it all in one person, but let's try. So she sent it out, basically, out to Seek, wasn't it? Is that where she found you,
that's what
Steve?
on Seek.
Oh, it was on Seek. And she presents me, she said hundreds of people came back to her. But she said out of those 100, she got 6 that she liked, and out of those 6, she gave me 2 to interview. Yeah, so this was
How
for...
good is that? That's really good about having an agency that gets you, though, isn't
Yeah,
it?
and she does get me. She's
absolutely.
been with me from the beginning, and so she got me my first person after Annika, and then it went from there. And Annika now works for Carren. So
And don't think you're getting her back either.
Did you scalp her for me? Headhunt.
Wow. I don't even know what to say to that. That's so hilarious.
That's what I know. It's just because I don't know recruitment, and Karen's very good with recruitment. But anyway, so I looked at both of the sheets, all the references, and I looked at everything that was, and I saw Steve's, and I'm reading through it, and I'm looking up at Sally going, he has everything. He's perfect. And you know what, Steve? And I know it was so funny. I'm like going, yes, yep, I want to interview him. And so what was really interesting is that I only wanted to interview you. I decided that I really didn't want to interview anybody else because I knew you were the right one. And the other thing was that everybody else was females and you were the only male. And that was something, and I don't know why I wanted a male, but that was one of the things that I wrote down, was because we have so many girls in our office. And I thought it would be a really good balance to have a guy in the office that could, keep Howie company and keep Tony company,
And keep you girls in line.
Yeah, keep us in line. So I interviewed Steve, and Steve walked out the door, and I said to Carren, I went, that's it, he's the one. He's got everything that I want. And I saw you, as soon as I saw you and you left, Steve, I said to Carren, he's the Steve Irwin of biomedical science.
Ah, look out.
Yeah, he's the Steve Irwin of biomedical science, because I knew Steve really well. So I then interviewed the second girl, and both Carren and I just looked at each other and went, no, we know who we want.
Did you have Carren sit in with you?
Always. Carren always sits in with me, and
that's
so does
awesome.
Howard.
Oh, yeah, cool.
Yeah, no, we always sit in together because we all have to work together, and it's always good. It's it's not just about me, although we like to think it is about
Well,
me.
actually.
Yeah.
And Obi was there as well.
Oh, Obi!
Obi was there.
Yes, Obi is a really
And that's
good...
what clinched it for Steve.
Yeah, Obi just turned on his
rolled
back
over
and
on
went,
his back.
I love him.
Stroke his stomach.
Yeah, yes. So what...
He's
accepted,
the
I
chief
think.
executive officer. They just don't know.
Yeah,
right.
he is the CEO, I can tell you that now. So anyway, what Steve's role in our business is head of education. Although that is coming. What he is right now is he's just creating the course for...
GDB,
Yeah. What's that?
General Dog's Body.
GDB, do I not know that term? So, and so what it is we're creating the education course.
Which is so exciting.
Yeah.
We have
it's
been
absolutely
dying for this.
fabulous.
And what lit us up was how quickly you actually appeared. I shouldn't say quickly.
Yeah.
Cindy's been thinking about this for so long, Steve. I just want to ask you, how did you find yourself in this position to be ready to apply for this position? Like, what's the synchronicity and timing on this?
Yeah, I mean, it's a fairly long story, but I'll shorten it.
You don't have to on this.
We like good
So I
stories.
suppose a little bit about my background
Yes.
is I studied at QUT, Queensland University of Technology, where I did my Bachelor of Science. And then I went into an honours program, and an honours program is a 1-year program where you're basically just thrown into the lab and you research something. And so I was heavily involved in genes involved in prostate and breast cancer. So that's what my sort of honours work was about. And then after I finished that, I decided I loved that work so much. I loved the genetics, the molecular biology. I just wanted to work more on this kind of stuff. So I did a PhD in...
As you would.
As you do.
As you do.
Yeah. As you do. On the genetics of endometrial, prostate and breast cancer, just looking at markers to see if we could discover any particular markers for prostate. Because the current markers for prostate and breast cancer are good, but they're not perfect. Because, for example, if you look at prostate cancer, one of the markers for prostate cancer is prostate-specific antigen, or called PSA. And you can measure that in your blood, but a lot of athletes, and PSA is actually regulated or switched on by testosterone. So a lot of athletes have high levels of PSA. So it's not a really good mark. It's okay, but it's the gold standard at the moment. So our role was to try and identify better markers. So that took me...
And did you?
Work is still ongoing, actually,
Okay.
but I've sort of moved on from that work.
And that was your PhD, that's what
I
you
did
did?
my PhD, but during that time I was really interested in steroid hormones and the way they actually switch on and switch off genes, because that's the work I was doing with prostate and breast cancer, because they're hormonally regulated.
Can I just ask,
Yeah,
quickly,
sure.
how old were you doing all this?
I was 30.
So you had
I
gone
started
from
a little
school.
bit, I started a little bit later.
Okay.
I was actually a refrigeration and air conditioning mechanic before I started all this. No, it's serious.
So that's a trade?
Yes, I left school and my father said you need to get a car, a girlfriend and a trade. So I got a car and a trade and then a girlfriend. Something like that. Yeah. So I did a trade for a little while and I thought, I've always wanted to study and I wanted to do marine biology. And when I went to university, the career counsellor said to me, forget it, there's no jobs in marine biology, you're better off to do biotechnology. I thought, okay, I'll do biotechnology. So
And that didn't scare you, just the word?
I was interested in science anyway, so I majored in
like?
biochemistry. And
Good question, Kimmy.
And if you could see Kim, she's leaning right into Steve. She's got her head on her hands, and it's almost like she's hypnotised. She goes, what's that like? To be interested in science, what's that like? It's like she just was totally lost in a moment. That's hysterical.
oh, that's funny.
Carry on.
Oh, and back to it. Okay, carry on, Steve.
No, that's fine.
I just love people like you, Steve.
Yeah, so I majored in biochemistry and molecular biology, and then that took me through to the PhD. And when I finished my PhD, I was really interested in steroid hormones and how they're involved in gene regulation. And I started to get interested in diabetes and metabolic disease, because metabolic disease is very hormonal as well. And so I moved to...
What's metabolic disease?
Okay,
Sorry.
metabolic disease is, sorry to throw that at everyone, metabolic disease is actually diseases such as diabetes, obesity, all of those, metabolic syndrome, all of those diseases that come under diseases involved in metabolism. So changes in your metabolism that cause disease. So, and diabetes is one of the most classic. Diabetes and obesity are diseases of metabolism. So
Really?
somewhere in metabolism, some part of metabolism is...
Because of what they're eating, it changes the metabolism?
it's a little bit more complex
I was
than
going
that.
to say, because all I'm aware of is you've got a fast metabolism, a slow metabolism. You know how people talk like that and blame that.
Yeah, I mean, if you look at obesity, for example, obesity is biochemical, which was metabolic, and then it's behavioural, because a lot of obesity is due to leptin resistance, which is a biochemical phenomenon.
What
But
comes first?
And then it's behavioural after that. What comes first, the chicken or the egg? That's a
Yeah.
hard question because we don't know. I mean, people are addicted to food these days. There's food everywhere. There's high sugar contents. Let's face it. Food tastes pretty good when it's bad for you. That kind of bad food tastes pretty good. So you get this kind of pattern of eating, and you want more and more. And we know now that sugar is addictive. So that creates problems with your metabolism. So whether that sets up a leptin resistance or not, we don't know. Certainly it sets up insulin resistance. We know that for type 2 diabetes, for example. But we don't know whether it sets up a resistance for leptin or whether leptin resistance comes first. So it's hard to say.
And we have talked about leptin resistance. We did a whole podcast on that.
And so then I moved to the US for a little while, and I was in the Baylor College of Medicine in Texas, and I was working there on progesterone regulation in breast cancer with a woman by the name of Nancy Wagle. And so I stayed there for a little while and moved back to Australia. I was only there for not long. Moved back
Houston?
in Houston, Texas. Yeah.
How'd that work out for you?
Give me speed to Houston.
I can see why you
I
came
don't want
back.
to offend anyone.
Yeah, no.
People from Houston listening. But
And it's a
Houston's
beautiful place.
place.
But no one lives in the city.
No,
How interesting
that's
that.
right. Yeah,
And
no.
the food is a shocker.
Yeah.
Let's just say.
Well, the steak is a massive.
Yeah, no, but
It's
for us non-meat eaters, not appealing.
No, that's right. That was a long time ago.
I can see where you came back, but
Yeah. So I came back and I worked. I was really lucky to get a scholarship. Not a scholarship, a fellowship from the University of Queensland, and I worked in a lab looking at metabolic disease, particularly type 2 diabetes and insulin resistance in skeletal muscle. So because of my interest in hormonal regulation, which my interest in hormonal regulation, I basically just fell into that job because I was really enjoying that side of it. And then I got involved in insulin resistance and type 2 diabetes. And I worked there for 6 or 7 years, basically, as a research scientist, writing papers, writing grants and trying to get more money to do the research, before I moved on to help set up a science lab at the University of Ballarat, which is now Federation University of Australia. So I moved there to help basically boost their research capacity. At that time, it was really quite small. So I moved down there for 2 years on a 2-year contract. They sort of, as Cyndi says, scalped me. Yeah, that's right.
It'll be a new word. Everyone's going to say scalping now. In Ballarat.
which is where you... No, you were Bendigo.
I'm Bendigo.
Okay.
Yeah, I was from Bendigo.
Still Victoria.
Yeah, we're getting closer.
We're getting closer.
We're synchronicity
Yes, I'm just
happening.
seeing here.
Yeah.
So I helped develop that, and I've actually got PhD students still working on a project down there involved in zinc transporters, and these are transporters that transport zinc into and out of cells, and their role in type 2 diabetes. And there's a lot of research on that at the moment. It's kind of a really hot topic. So I've got a couple of PhD students working on a project down there that I collaborate with externally. I meet up with them once a fortnight via Skype or something.
I just need to ask this question. So I get really excited to get up in the morning to go for a run.
Right.
And I get really excited to make beautiful food. And I get really excited when I'm even writing a newsletter and things. But what happens when you are looking down a microscope? I mean, do you get that same adrenaline?
It's true. I do. It's
I'm
crazy.
hearing you.
I mean, and a lot of the work that I do is...
You are hilarious.
Yeah, it's really true. I mean, it's so funny because most of our work that we do, we're passing one colourless liquid from one tube into another.
That's right. So that's
And
not
that's-
just for show on TV when we see them doing
well,
it.
they put a food dye in that.
Right.
And they hold it up like they're doing
Right.
something, but it's just water and food dye. We've all done that.
Okay.
And
That's a good... I didn't know that.
yeah. Because you don't want anything toxic, especially around cameramen and stuff
Oh,
like
that's
that.
too funny.
Yeah, and they have to come into a lab that's, restricted. So most of the time, it's just the colourless water with
food
colour dye. So, but when I get really excited about the science, when I see the experiments that I'm doing, and I'm taking the research that other people have done, and I'm repeating it, and I'm trying to add on to it, and think of my own ways that a particular system or a mechanism might work out, and and I can achieve that, that makes me really excited. And just to look down a microscope, now microscopes aren't like you're just looking down them like the old days. Now we've got them on big screens, so we've got huge kind of instrumentation set up where we've got screens, massive screens, and you can put your slide and your piece of tissue or whatever you're looking at under the slide and just view it on the huge screen.
Wow.
And we can do it in 3D now, so we can take slices through cells, so we can have basically a bit of tissue sitting on a microscope, and we can slice through it in silico, which means in a computer system. We can slice through it in a computer system, take slices through it. So, and these are like a 1 micrometer slices. So you can imagine 1 micrometer is 1,000th of a millimeter.
Oh my
so
God.
you can slice through the cells and then take shots.
I can see this.
And so you can see right through a cell, and you can see where different particular parts and components of the cell sit and how they're interacting with certain drugs that you might be putting on the cells or so forth. So it's pretty exciting stuff.
Do you look at people, like seriously, do you look at people and go, oh my God, if only they realised on a cellular level what they're doing every time they, like, you know how hairdressers go, oh, I wish I could do something with their hair. I mean, are you looking at them and going, I wish I could do something with their cells? Seriously. Because if you look at people on a cellular level, everything, like I'm just aware of everything that I eat and drink and touch and do, like everything I put on my skin. You're just looking at your hand. You're not going into millions and trillions of cells. Like, do you think like that?
No.
But just well, I'm not a scientist.
No.
The pressure's off, love.
No, I don't really. I mean, I'm overwhelmed and in awe of a cell and what it can do, and the things that we still don't understand.
That's what I was going to ask you. Like, do you find yourself in awe of the intelligence that exists inside of a cell and how it does it?
Yes, I do.
Did any of your studies include, because I'm the other side of that, so do any of your studies include that innate intelligence that exists inside of the...
Yeah, that's a good question. And, well, there's the difference as you're looking at the vitalism side of things and the innate ability of a cell to control its environment or to respond to
how does that
some
respond
sort
to
of
a
external
drug as
stimulus,
opposed to...
for example.
Yeah.
I mean, and then there's the reductionist theory, which a lot of scientists sort of, are really sort of intent on studying, taking that one component of a system, studying it in detail, which is very good because you can work out mechanisms, and that's why how a lot of our drugs have been designed today for things such as, for example, schizophrenia or type 2 diabetes. They're all designed at that level of the single cell before they're put back into an animal model, tested and so forth. So you really need to understand those mechanisms, but that's not the whole story, because we know that there's other things that are happening within a cell that we don't understand and we'll probably never understand. The ability of just your body to heal itself naturally. I mean, where does that come from? And, people say, well, it's all biochemical. Well, that's true. It is biochemical to some extent, but how does that start?
Yeah, where's the source?
Yeah, so I think there is a bit of a shift in the way that scientists are thinking now, particularly in biomedical science. It's starting to move that way because the emphasis is on taking the biomedical science from the lab to the bench, from the bench to the bedside. Being able to take the work that you're doing in the lab and in the context of taking it into a bedside in a hospital, for example, rather than just looking down the microscope, as Kimmy was saying this just before. So I think there is a little bit of a shift in understanding that we are just not we're not just the sum of our parts. Basically, we're more than that. We all interact with looking at the whole system.
I am actually fascinated by these. I'm loving it. Would we be able to come into a lab with you one day?
Sure, you could.
No, I'm serious.
No,
I'd love to see it.
yes, for sure. If you wanted to come into a lab,
We would film that podcast.
I
reckon that would be a video podcast.
But remember, it's only going to be water and coloured dye.
I can show you some cells though, some living cells under a microscope and things like that.
That would be amazing.
Would that gross me out?
No, not at all. No.
So can you see that? Because she does get grossed out very easily.
No, you wouldn't be grossed out. No.
So can you see the mitochondria? Can you?
No, you can't.
You can't.
You can't.
That's what I want to see.
You can. Depends on what you're using. Just if you're using a normal confocal microscope, which is a kind of very standardised microscope, you can see cells and you'll be able to see the nucleus, for example, which houses the DNA. And you'll see... And what's really interesting is I used to work on skeletal muscle cells. And skeletal muscle cells start in what's called myoblasts. So they're not really skeletal muscle. They have to be induced to become myotubes, which is skeletal muscle. So the microtubules that we have in our muscle. So we induce them with various hormones, which is what occurs in our own bodies. But when they actually become myotubules, they become very striated. So they're actually like long filaments, and you can see them actually contracting under a microscope. So that's pretty cool when you see that. But you can't see individual mitochondria and other organelles because they're too small, but you can with an electron microscope. So if you use an electron microscope, which basically fires electrons at the organelles within a cell and forms an image. It's like similar to an X-ray diffraction or something like that.
And does that actually, when you fire those electrons, does that actually affect the matter?
Yes, that's a really good question. There are ways to actually get around that. So that's one of the things that when you're looking at when you're doing microscopy, that kind of work, you have to be really careful that the things that you're doing, the way you're preparing your material, is not actually affecting the actual material that you're preparing. So that's why you have to set up controls. So you always have controls, and that's one of the big things. So yeah, that certainly does happen, but it's with our technology today, it's very minimised.
I can ask him anytime. You girls go.
So when my husband played cricket professionally,
right,
and he did a hamstring tear, and
right,
we went in to have an MRI scan,
right,
and I watched with the radiographer how they sliced through.
Yeah, sure.
The images of it looked like a leg of lamb sitting up there to start
definitely,
with,
yeah.
And then they went through each layer level, so that was on a muscular level,
right.
When you're looking at it on a cellular, it's just quite overwhelming for me to imagine, because when Carren's often taken us down the rabbit hole about from a microscopic or a macroscopic effect on our intelligence, if you like.
Yeah.
How do you not let emotions come into something so scientific and black and white? You know, like, does that ever happen in the sense of, oh my God, this is exciting, and then are you making something happen? Like, as a scientist, are you really having to be black and white the whole time?
Yeah, sorry.
I'm just thinking my question's not dissimilar to that.
And mine wasn't either, actually. That's so
funny.
funny.
It's funny.
Yeah.
Because what I'm thinking is, everything that I read, it says that the observer affects the object. And simply through the observation of an experiment, it affects the outcome.
Yeah.
And what you're suggesting now is, like, your emotion, does it actually affect how you read it? Okay, over to you, back to you.
Yeah, it's a good question. And it goes back to Schrodinger's... Schrodinger was the famous physicist that did the cat in the box. Schrodinger's cat. Have you ever heard of Schrodinger's cat? Where he put a cat in a box, a thought experiment, and it had some cyanide in the box, and a certain thing would trigger the cyanide and kill the cat, but you wouldn't know if the cat was dead or alive. So in a sense, it was dead and alive at the same time. The only way you could find out was by observing it, and then you change the whole parameters. So by observing something, you changed it. So that's very similar to what you're saying, Kimmy, and what Carren was thinking. And also what all of you were thinking about.
Particles and waves experiment with it.
Yeah, I mean, it's something that as scientists we do get excited, but we really need to be black and white, because we really need to understand, is this what we're really seeing? And that's where the literature is really important, and that's where peer-reviewed literature is very important. So when we publish a paper, the last paper I published last year went into a journal called PLOS ONE, which is a really good journal, but it came back 5 times before they were happy with it. It went out to 3 reviewers in the field, really high in the field. They reviewed it. 1 reviewer just was not happy at all. So we came back, we did some more experiments to satisfy that reviewer. It went back out. That reviewer still wasn't happy, and then sort of basically left the review panel. They had to get a 3rd reviewer. Then finally, fourth time lucky, we got it through. But it's just
What do you mean they don't like it? Is there something
it's not that they don't like it. They question something about it. It doesn't stand with their philosophy on how things work, maybe, or their understanding on how things work. So they, take a different view of what you're doing. So it goes to another review, and then it just sort of cascades on. And it might take 4 or 5 times to get something published, but once it's published, it's really been through really hard, stringent control through peer review of scientists that are really high in the field. It's not to say that it's actually perfect.
I wanted to ask
it's
you
still open to perception.
It's still open to perception.
But I also wanted to ask you, and tell me if I'm wrong, but
Yeah.
you can almost prove anything to be right or wrong.
You can, and that's where it comes back to using the theory that's available. It's really important to understand theory, and it's really important to understand it at a really deep level, at a molecular level. No matter if you're working in this field of biomedical science or whatever you're working in, you really need to understand it at that level, because at that level is where everything has come from. All of our, Watson and Crick discovery of DNA and all of that, it's all come from that foundation. So the foundations are really important. Not to say that they're exactly right, but if you can experiment enough.
What about the power of persuasion, though, from a marketing perspective, with a company, like a food company, that could employ... Are you guys bought? Can you be bought?
Can we be bought? Well, yeah, pharmaceutical companies buy us. Yes, definitely. They do. I would certainly not work for a pharmaceutical company, because I enjoy science for what it is in its purest form. I love science. I mean, I just love doing it. I wouldn't sell myself out to a pharmaceutical company. But people do it, and it's a lucrative career for them. But it's just not what I would do.
Depends on what the drivers are.
Yeah, exactly. Yeah. And science to me is not about money.
So I've got a question that I've always wanted to ask somebody like you.
Yeah.
Will you go out with her?
Oh, you are on fine tune, Kimmy.
Very on fine.
She is. But this is what she's usually like, I have to tell you. Mr. PhD.
I've only met Kimmy. It's the second time now.
She flew into the office.
She's grown on me very much.
I love you. And you're in my bedroom.
Yes,
I feel there's bonding going on.
Right, go for your question, Carren. I'm hanging out for this one.
Well, it's not anything that you guys don't already know, but like every time I'm doing anything, because I'm a bit of an analytical creature, so I really do like to go down the rabbit hole, and I really do like to understand the science of it. Yes, I find myself... Well, I find myself equally drawn to the psychology or the intelligence of it, and also to the scientific aspects of it. I love it. So when we say, and please forgive me because it's probably going to be the most stupid question for somebody like you, so please let me apologise in advance.
There's no silly questions.
Oh, no. Hi, I'm Carren.
And I'm a silly question asker.
I'm an Aquarian, and and we go out with me. Stop it. So when I'm doing all of my reading and I'm doing all of my research and stuff, and I've got like stacks and stacks on quantum physics and quantum mechanics and all of that sort of stuff. The question that I have is that when we're looking at things scientifically, and even just the body, when we're looking at the body scientifically, and science is very black and white. From all the research that I see, it is very, very black and white when it makes its assumptions about what the body does and it doesn't do, and its theories. How can we suggest that a theory is as close to being right when the source of its performance is undiscoverable? And I know that there's lots of studies on consciousness and lots of studies on awareness and all of that happening now, but even there's still no answers on that.
Yeah.
So it's almost like saying, well, the body is a physical being, and that represents, let's say, 50% of what actually is. So the finger, it's physical, but then there's an intelligence inside of that makes it function as a finger and keeps it alive as a finger that science actually can't measure. Yet the scientist will say, I'm aware, and I know that I'm aware because I wake up every morning with myself. I'm aware, but because I can't measure that, therefore it's not real. So how do we say that the basis of any theory that has anything to do with the body or life or humanity, or whether it's fish or trees or whatever, how do we actually get to claim any kind of theory as potentially the platform that we then go and do all the rest of our research and create our medicines and our responses to the world, when really only 50% of the understanding is present?
Good question.
It's a good question.
Because I've always looked at it and I thought, how can anybody say that? That's right. I just don't get that.
Yeah, you're right. Yeah, you're exactly right. And I was telling Cyndi the other day, and I've done all the statistics on this. About 90% of the research that's out there is not. You can't repeat it, basically, because it's been done in labs and they've used their own particular resources. And if you take that same experiment and do it somewhere else in another lab, down the road or overseas, 90% of the time you
replicate
can't do it. But that doesn't mean it's good. That doesn't mean it's no good.
Yeah.
Because what it does is it allows us then to go in and see what has been going on with those particular experiments, and that can help us build from those. Then maybe redesign the experiment. So all of that work is not to waste. And then there's only 10% of the research out there that's actually gone on to, send us to the moon, to, design iPads or whatever it may be. All that research that goes into keeping us alive in medication, for example, is the 10%. The other 90% is really stuff that's out there that just adds to the jigsaw puzzle. But the scientists that are actually doing the 10% of the work are going and reading all those papers to find out what's going on. So the idea of the theory thing, it is a theory, and it's open to discussion. And if you look at the classic theory of Lamarck, when he was discovering the theory of evolution, he thought that giraffes evolved long necks because they had to reach up higher to grab higher leaves in the trees, and then that was thrown out by Darwin's theory, which sort of surpassed that. But now it's coming back to Lamarck's theory because of the epigenetics that we have. So theories are always open to discussion and debate, and they'll always be surpassed. There'll always be another theory. So I don't think you can say that, even though we work in black and white, theories are never set in stone. They're always debatable, open to think. And that's the way the world works. We build on what we know and what works at the time.
So would you say you guys are theorists rather than scientists?
I think it depends on your discipline. Depends very much on your discipline. Biomedical science, we're probably more in scientists trying to understand mechanisms, which don't really build to a theory, but they just try to understand.
You just said then trying to understand, so therefore it's not black and white.
Yeah. Sorry. Yeah, no.
She's saying you're not definite, you're not black and white, and you're saying trying.
Oh, yeah, well, we're trying to understand something, but it'll always be right in our minds, or not right. I shouldn't say right in our minds.
One way or the other.
One way or the other, but it's always open to discussion. That's what I meant by that.
So, in when you say so, it depends on your discipline.
Depends on your discipline.
There are some of you guys who are simply just studying the physicality of it.
Theoretical physicists, for example, is a classic example. They just work on theory, and we're more practical scientists, I'd say, more what they call basic science, where we work on mechanisms to understand things. They don't necessarily.
PhD is basic science. I love it.
They call it. I don't know whether that. I like that word, basic science, because it's not basic at all.
No.
It's but we never work towards a theory or anything. But we try to discover ways things work, which then are part of a huge, conglomerate of components that add to the jigsaw puzzle that people can then just work on, and work that I've published before, people are working on it and trying to build on it as well.
Steve, my grandmother, when she was 93 and she had a stroke and she was in hospital, and my grandmother used to take 13 pills a day, I think 2 or 3 times a day. They had their little containers and everything. She had a stroke, she was put in hospital, told nil by mouth, was taken off everything, and we all came in to say goodbye. My grandmother got well enough to immigrate to Australia. At 91 years of age, she was 91.
Wow.
What fascinates me from a science medical point of view, or a drug point of view, is one drug is made up of all these things, and they give it to that person. Then because of that drug, it upset her tummy, so they gave her another drug. And then she needed warfarin for her heart, and then she needed an anti-inflammatory. How on earth do you guys work out the effect of now 10 different or 13 different tablets on one person? But my question is this. She was taken off everything. We were told to say goodbye. She got well on no medication.
Yeah.
And my thing was, why would we put her back on it?
Yeah, exactly. Why would you? Yeah, when she's well.
How do you know, though, that 13 tablets in that particular person's body is not going to cause her... is it just all trial and error?
Yeah, it's the whole idea now with drug therapy is to really go to that silver bullet where they're trying to personalise medicine. But it's a really hard sell because we're all so different. We all respond differently to different drugs. And we know that even with drugs for diabetes, people that are diabetic respond differently to different drugs. For example, metformin, which is a drug used for type 2 diabetes, people respond to it and they have different side effects. So the idea of understanding drugs in a system is really looking at a very broad sense on how it interacts with its molecules in itself. how it interacts with receptors and what it does. But then putting it into a person, for example, is going to be a different story because we're all very different. And how it acts in that particular person might be different from another person. So it's really difficult. So most of the drugs that are designed on the market now are designed over a broad population of people that are, for example, type 2 diabetic. So they design drugs within a system, they go into clinical trials over a population of people, and they all have different side effects, but they kind of live with that. So the idea of what you were saying is, how do you understand 13 different drugs and the different side effects they'll have?
The cumulative effect.
The cumulative effects, that comes down to just basically clinical trials, and there's a lot of drugs that have been taken off the market because of severe side effects.
When I was in Houston, nice place.
Yeah.
There was an ad on TV for Viagra or some sort of Viagra, some sort of, I don't know, sexual enhancement, and then it had all these side effects come up underneath the ad.
Isn't that hilarious?
And one of them was impotence.
Yeah.
And I'm just thinking, so when you, I don't know, I just find it very interesting from a pharmaceutical point of view, and then you had the slant on it about, that this is going to make you feel good and it's going to do all these different things to you, and then one of the other side effects is it could kill you.
Yeah.
So, yeah, cause nightmares, high blood pressure, suicidal thoughts. Don't take
Depression.
Contact your doctor if you have a skin lesion. There's
It
all sorts of things.
It's interesting
So
because
they cover themselves well.
Yeah, it's interesting.
They do, don't they?
If you take, for example, type 2 diabetes, and if you're insulin resistant and you're type 2 diabetic, your insulin's not working anymore, so you're on sort of polypharmacy where you're on insulin and other drugs. There's other drugs that actually make you put weight on, which kind of
Obesogens, they are called.
Well, yeah, obesogens. You've done your research. That's very good.
Oh boy, if I could just have a little halo above my head right now, I'd be so happy.
I'm impressed, Kim.
But what I was going to say is, obesity promotes resistance as well. So it's kind of a vicious circle. So you're taking drugs that will enhance your insulin uptake, so that you don't have blood glucose, floating around in your system causing all sorts of problems. And at the same time, you're putting weight on, which enhances the insulin resistance. And then if you think about how insulin works as well, insulin basically inhibits the signaling of leptin to the brain. So you've got insulin and leptin working together. So
shut the front door. So what are you doing about that, Steve?
So there's some interesting work that's been done with obese patients that are not. So we're going a little bit off track here, not so much, but with obese patients that
I'd call us scientists.
yeah, that's what we do, don't we? Yeah, and
or theorists.
theorists,
yeah.
That leptin resistant. So leptin is really important. It's secreted by fat cells, and it acts on your hypothalamus to say, hey, I've had enough food. I don't need to eat anymore. A lot of obese patients, their leptin's not working. The signal's not getting to the brain, so they become leptin resistant. So they've found that actually drugs that are involved, and that's also insulin is involved in inhibiting leptin signaling in the brain as well. So you're also producing high levels of insulin because you're eating a lot of food and you've got high levels of sugar in your blood, so you're producing a lot of insulin. So insulin is having that additive effect as well as inhibiting any leptin that's trying to get to the brain. So they put obese patients on diabetic drugs to lower insulin levels, and they've had a response. So leptin can get to the brain, and they're not as resistant, so they start to lose weight. So when they put them on these diabetic drugs to actually clear insulin from the system, these patients lose weight. So
but then what happens to all this? Because they're still eating lots of sugars and stuff. So what happens to that?
This is on a normal diet. If they just eat what they want, as they always are, you can actually put them on diabetic drugs to actually increase their leptin sensitivity somewhat. And I'm sorry, I forgot where the question was at the start.
I don't know. Who cares? It's just fabulous.
Keep going, I think we were talking about
keep going.
we were talking about how drugs work in the systems and the different ways they work. It's so complex. We, as I said, they interact with
Russian roulette sometimes.
And some, like for example, the glitazones. These are drugs that are used in type 2 diabetes, which act on nuclear receptors, which are called transcription factors, and one of the classic ones is called PPAR gamma. You don't have to worry about it.
See how I don't go this deep. There's just no way. Like when when you were telling me this the other day, I'm like going, oh my gosh, like going through each layer of the cell to understand. Yeah. So sorry, Steve, I interrupt
that's all right.
you.
No problems. So those drugs that act on those particular receptors are involved in. They bind to the receptor. It then translocates across in the cell and gets into the nucleus where it binds to DNA, and then it modulates gene expression. So what it does is it binds to DNA and then it switches on or switches off genes involved in lipid metabolism, for example, in the case of the PPAR that I'm just talking about. So when you're actually looking at those kind of drugs that are targeting those particular molecules, they don't only just have that single effect by binding to a particular piece of DNA and regulating the gene for that DNA, they have multiple effects. So they bind to other DNA as well and have different effects. So those kind of drugs that we see now in society for type 2 diabetes, for example, or whatever it may be, have a plethoric amount of targets. So they target many, many signalling pathways, and that's where the problems start to come in, because that's when the side effects happen. So it's really difficult to single out a single drug that has a single target. It can be done, and that's through chemical modification. But that's the ongoing work at the moment.
And have you done then much work carrying on in this into the thyroid in relation to the way?
I haven't done a lot of work on the thyroid, no. Most of my work, no, I haven't, no.
I've done a lot more than me. But I'm just interested at the moment. The thyroid seems to be a massive topic.
Yeah, oh, it is.
He will be researching that. Don't
I
you
will
worry.
be. I can give you another update on that once I start
we'll get you back
researching
in a few months' time. Next week.
Yeah.
So can you? Oh, sorry.
I want Steve to tell you, Kimmy, about exercise, because you love your exercise, and I would like you to talk to Kimmy, because this is her baby. She's done 24-hour marathons. This one, she does crazy stuff like Kokoda trails. That's her whole life is this type of taking her body to the absolute extreme.
Yeah.
So can you talk to Kim and us about how exercise changes our gene expression?
Yeah, sure. So you've heard of the epigenetics. So epigenetics is changes in your DNA, but it's not actually changes in your DNA structure per se. It's changes in the way your DNA is switched on or switched off. So epigenetic changes don't necessarily need to occur in the particular gene itself. They can occur a long way away from the gene and still have an effect on the gene through various regions I won't get into in this discussion now. But so exercise can actually stimulate genes to be switched on and switched off. And I went to a conference in Japan last year called Diseases of Aging, Aging and Diseases of Aging, and they presented this work done by this lady presented this work on these athletes that were actually on intense exercise for 20 minutes, and they basically scanned the whole epigenome. So every single piece of DNA in your cell, every single gene in your body was scanned for changes in epigenetics. And what they found was that pre-exercise, 75% of the genes were basically switched off. So they show you this big heat map, and red means off and green means on, and 75% were red of the whole panel of genes. The 25,000 genes we have in our body that are at any one time switched on or off. After 20 minutes of intense exercise, it was 75% were green. So they're all switched on.
Wowza.
When they actually went...
20 minutes, that's all we need to do. We don't have to do our 24-hour marathon.
No, it's true. It's 20 minutes. And it doesn't even need to be intense exercise. And you don't have to be... These were athletes that were doing it, but I have a colleague back at Federation University that's also doing this work on people that aren't athletes. They just like exercise. And they've also seen the similar changes. But the changes in the genes that they saw were genes that were very much involved in energy expenditure, metabolism, things like that, things that you would necessarily need to sustain you through that particular exercise process or what you were doing. So in 20 minutes, you can actually change your whole genome. And all of these genes are genes that are switched on for good reasons. So they're reasons for burning fat, for supplying energy and all that sort of thing. So, yeah.
It's interesting because as a personal trainer, when I trained in the 80s and 90s, it was all around 1 hour of exercise, 3 45-minute weight sessions a week, and all this sort of thing. But lately I'm doing a lot of, the 20-minute intensity exercise is one of my favourite. Yeah. And all my WODs and things that I put onto my pages now are between 2 minutes and 15 minutes long.
Yeah,
and the results I'm even getting, and I wondered if that was because I was getting older, but then I'm noticing younger athletes are getting a much better reaction and better, not as many injuries. They're feeling stronger. You actually don't need to work the body out the way we used to think we had to.
No, definitely not.
I did a show on Carren's couch a couple of weeks ago with a man in the United States. He owns the Beverly Hills Anti-Aging Clinic. His name's Dr. Uzi Rice. And we were talking about lots of different things, but one of the things that he said was that...
Sorry.
What?
Uzi Rice.
Yeah.
It's funny, cha-cha.
Oh, yeah.
What? What?
Steve worked with a professor named Dr. Funny Cha-cha. And when you said Uzi Rice, all I could think of was Funny Cha-cha.
Funny Cha-cha?
I love these names.
Yeah, that's why I'm not a scientist.
You don't have a good enough name, that's what it
What's
is.
your last name, though?
Morrison.
Oh, well, that's a good sign.
Sorry, I just...
that's
Uzi
hysterical.
Rice. Sorry, go on. We'll never forget his name.
Said sushi rice.
Oh, don't. No, the man's a genius.
Okay, all right. I know they might have funny names, but they are geniuses.
I know.
Yeah.
But he was actually saying something similar. He was saying that when our bodies respond far better from an anti-aging point of view when we do the short, sharp 2, 3, 5 minute max and then rest for 6 minutes and then 2, 3, 4, 5, 6 minutes max and then rest for 10 minutes. Like he said, we've got it the wrong way round in order to switch on the anti-aging.
That's why I think CrossFit and things like that have become so popular. They're quick, short, sharp, intense exercises. So from an epigenetic point of view, I wanted to ask you something about, is it the BRCA gene, the breast...
The breast gene.
BRCA1 and BRCA2,
Let's talk about that because
can I
I've got
just-
somebody who...
I just wondered, you were saying before the genes switch on and off, the 25,000 all at different times.
Yeah.
So what's your thoughts around women that... do we all carry the gene? And
Yes.
therefore...
Oh, we all carry the gene.
Well, so we all have that gene in our DNA, but it's actually...
even men?
We all contain the gene. I'm not sure. I'm not sure about men. I don't know about men, but we all
Because men
we
can
all
get breast cancer, can't
we,
they?
we can, but I'm not sure whether it's related to the BRCA1 or BRCA2. I'd have to look at that. But it's actually a mutation in that gene that causes causes the problems. So if you're screened, when you're screened for BRCA1 or BRCA2, you're screened for the gene, but you're screened for the mutation that it carries.
Is
And
that
then-
why women then remove their breasts?
Yeah,
Have they got that mutation?
Just in case, because it can lead to. It may not, but it may.
So
it's like
have they got the mutated gene?
Yes,
Yeah,
they would
if
have.
they come up as positive
if they
with
come
the
up
BRCA2.
positive.
Does it mean they'll get breast cancer though? You're saying no?
Not necessarily. It depends. Some women just opt to have it removed just in case because they have a family history of breast cancer.
So
But
if
what
they do
makes-
have a family history of breast cancer, is it more likely that will develop into breast cancer?
It probably is more
It is,
likely,
okay.
yeah. So the BRCA genes are genes that we have in our body, the breast cancer genes, but they're they have normal functions. They actually have normal functions in the breast, but when they're mutated, that's when the problem occurs.
So how do they get mutated? Is it from birth
It's-
or is it an
well-
epigenetic thing?
Well, with these genes, BRCA1 and BRCA2, it's heritable. So it's passed on through generations. It's not that I know of. It's an epigenetic phenomenon.
But then it has to be turned on, right? Because genes are turned on and off, as you were talking about.
Normally, so what happens with genes is when they're mutated, when genes are mutated in certain ways, they either are not made into proteins anymore. So if they're not made into, so say there's a particular gene X, call it gene X. It's made into a protein, and that protein does something in the body. If it's mutated, it may not be made into a protein anymore, so therefore it won't be able to do what it normally does, and then that's where problems start. Or the mutation will occur and the gene will still be made into protein, but the protein will be misfolded somehow. So it won't fold properly because it has that mutation, so therefore its effect will be somewhat diminished. It may still have a slight effect because it's still got a similar structure to its wild type, which is its normal protein, without the mutation, but it will actually be less efficient at what it does. Or it could be just totally non-functional at all.
Or dysfunctional? Can it do something it shouldn't do?
It can, exactly. So if you have a protein that's not folded properly, it could bind to something else that fits its kind of shape and switch something else on, for example. So that's how mutations generally work. They're either not made into protein because the genes are switched on and they're made into DNA, into messenger RNA, then goes into protein. And if there's a mutation that stops it being made into messenger RNA, then it won't never make protein. And then having it lost from the system could cause huge problems if it's something that's really required. And then the other option is that you could have a mutation which will actually make more of it, for example. So you'll overexpress it. So having too much of it can be a problem. Or you have a mutation where it will make messenger RNA, then make protein, but the protein is somehow, its conformation is not right. So therefore, if you look at proteins, the way they fit in with certain other proteins is similar to lock and key sort of style, and it won't be particularly be able to fit a certain other molecule or whatever it may be binding to, and can cause effects that way. So that's how mutations work. But epigenetics is different. It doesn't work based on mutations. It works based on chemical groups that bind to DNA in long stretches on the DNA at their gene level to switch them on or off. So it's actually not a physical mutation in the DNA itself. It's something that binds externally to the DNA to actually block it.
What's the difference between DNA and RNA? You're talking about
DNA and RNA. That's so we all have. So DNA is we all have
deoxy nucleic acid.
Yes, right.
Deoxyribonucleic acid, isn't it? What
Perfect.
Is it?
Yes.
Oh, well done. Tick, 100%.
Yeah. So
what is the difference?
DNA is in all of our bodies.
Yeah.
For every single gene, we have the DNA in our bodies. I mean, the DNA in your finger, for example, contains the DNA for your colour of your eyes as well. Obviously, it's not switched on in your finger. It's switched on in your eyes. Okay. The messenger RNA is the genes that are switched on. So you have the whole complement of genes, 25 of your genes in your whole body, 25,000 of them or so, and the ones that are switched on at any one time is the message RNA. So, for example, in your eyes, you'll have the gene for for prostate growth or something like that, or whatever it may be. But it's obviously not going to be switched on in the eyes. You know, it's going to be switched on elsewhere. So, but the colour of...
Hopefully in the prostate.
Yeah, hopefully in the prostate. Well, you don't want your prostate to grow.
Yeah, you don't want one of those. So what's the difference then? You said that somebody who does have that BRCA2 gene, or the BRCA gene, you say that that's hereditary. So that's what's likely to, if they do have that in their family, and then it could cause the breast cancer. But what if you do have it? And it's not necessarily a determinant or a precursor to cancer. What are the conditions that has it not be a precursor to cancer? So if the conditions that has it be a precursor to cancer is that it's hereditary, what's the conditions of it not being a precursor?
Yeah, it depends. It depends on. So it gets down to the mutations in the BRCA gene. A lot of genes have many mutations. I was telling Cyndi the other day, the estrogen receptor, for example. If you just take the estrogen receptor gene, there's something like about 8 different mutations in it, and the estrogen receptor has been well known to be involved in breast cancer as well. So it depends on the severity of the particular mutation they're looking at. The BRCA gene might have other mutations that aren't even discovered yet, but that they know of in certain populations, but it may have no effect. But if it's a particular mutation that's well known to be a precursor for breast cancer, then that's the issue. So it'll be well known.
We've got somebody that we know that has been assessed as having the BRCA2 gene. Yeah. And she does have family members who have had breast cancer, and she doesn't know what to do next.
Yeah, and it's because if you think about the when I was just talking about the message RNA and things like that, because that's the particular gene that's in the breast that's switched on in the breast. Yeah, it's not switched on anywhere else that I know of to do anything else. But it depends on the particular mutation. So, and I'm not an expert on the BRCA1 or BRCA2 gene, but there'll be a mutation that's involved in that's actually a precursor to breast cancer that is well established. And that's what they'll look for. When they screen for you, they'll look for that mutation. It's the same as cystic fibrosis. There's a delta 508. Delta 508 means the delta sign is a change. In 508, it's that amino acid, which is amino acid is part of a protein. At 508, there's a change in that particular protein, and they know for a fact that that's the change in the protein that is involved in chloride transport in cystic fibrosis patients. Right. So they would screen for the delta 5/8, and if you have that delta 5/8 change in 508, then you could be a carrier, or if you actually got both alleles or both genes carry the same mutation, then you will have cystic fibrosis. So they will screen for that particular mutation. The cystic fibrosis gene also has many other mutations in it, but they're not a precursor for cystic fibrosis.
Right. Okay.
But in saying that, they're kind of additive as well.
Well, we hope that you've enjoyed this interview as much as Kim, Cyndi and myself have. Talking to Stephen is just like, sitting in front of the guru, the font of knowledge. And how lucky are we that we get to have him right there in our own hip pockets. Woo! So go ahead and tell us what you think of this interview. Go ahead. You can type your comments at allthews.facebook.com/up-for-a-chat, or you can also go to allthews.thewellnesscouch.com/up-for-a-chat and join us here next week and become part of the ripple effect that's changing the world. We're going to see you on the ride.
Hi, Brett Hill from the Wellness Guys and That Paleo Show here. We had some incredible feedback last year that being at the Wellness Summit with a group of like-minded people made it just that much better. So this year, we've created a special group rate for 10 or more people to attend the Wellness Summit. For spreading the wellness message, you can come to the summit for just $197. That's a saving of $100. And all you need to do is get you and 9 other friends along to Crown Melbourne on Saturday, August 16 and 17 for this once-in-a-lifetime event. To book your group tickets and have the best time ever with your friends, go to www.thewellnesssummit.com.
This has been a production of TheWellnessCouch.com. Check us out on Facebook and join in the conversation on Facebook.com/TheWellnessCouch. Subscribe to each show on iTunes and check us out on Twitter. The Wellness Couch, streaming wellness into your lives. Whilst the Wellness Couch presenter endeavor to provide accurate and helpful information to their listeners, these podcasts cannot take into account individual circumstances and are not intended to be a substitute for health and medical advice from a qualified health professional. You should always seek the advice of a qualified health professional before acting on any of the information provided by any of the Wellness Couch podcasts.